Hello everyone. Although my last experiment kind of didn't really flop, because that's not really how experiments work, I guess, but as long as you do everything right. But my indole acetic acid treated algae ended up just completely dying within 24 hours. My Naphthaleneacetic acid treated algae, it did in fact survive. So what I'm doing now is I'm going to actually count them, and I'm going to do some measurements. I'm going to take a huge sample of measurements to be able to see if there's any changes within the algae, if they've grown differently. Or first, I'm going to fill this with some of the media that I use there as a as a standard measurement. Because before you can you can use this machine to actually measure, you have to calibrate it. Otherwise, you don't have a reference. And what I do is I bring the, because this thing reads out in millivolts, and what I do is I bring it down to, so that the reference is always 400 millivolts. So I bring down the intensity of the light. Okay, perfect. Right now I'm just kind of mixing things around to make sure I get a good sample. And I have a count of 357. Now that's, or not a count, but that's the voltage. But I have a I have an algorithm that I use to figure out the actual count. Here. And it's log 400 divided by 357. And then times 2083535. And that comes out to a 103,000. It's 102,990 or 909,000. 102,909. But this is only accurate to within 3 significant figures, so 103,000 trichomes or cells. So 103,000, 103,000 times 500. There's roughly 51 million trichomes in there, in this container. And then now what I'm going to do is I'm going to take a sample of this, and I'm going to put it under the microscope. I just need 5 microliters. I'll take a picture of that one. That's definitely a misshapen one, because one of the issues that I've seen in some papers is that these hormones can actually cause morphological issues, which is just a fancy way of saying misshapen growth. Right now I'm taking a bunch of pictures. That way I can do some measurements. So far, other than those couple, I haven't seen any more misshapen ones, so I don't know if that's just an anomaly that has nothing to do with the actual effects of the hormone or not. So the way I measure this is from tip to tip, and of course I only measure the ones that I have in full view. And what I'm going to do is I'm going to compare these to a database that I've created that has about 1,500 measurements in a population, in a spirulina population. And the idea is to see if the hormone in any way changes the life cycle of the spirulina. Okay, now I'm not going to bore you with 500 measurements like this, so I guess I'll check back in later.